Good evening, everyone. So, I am Vineet and I am your host for today. So, let me introduce you to our speaker for the day, for the session. So, it is my pleasure to present Surajit Malakar, a PhD student at the Center of Genetic Disorders, Banaras Hindu University. So he is working under professor supervision of Professor Parimal Das, and his objective is to uncover genetic variations related to Parkinson disease in the eastern part of the Uttar Pradesh population. And he has a deep interest in the genetics of neurodegenerative disorders, particularly movement disorders such as Parkinson's disease. And today he is going to take a session on exploring genetic basis of neurodegenerative disorder, taking Parkinson disease (PD) as a model. So uh, I welcome Surajit Malakar.
A very good evening to everybody. Hi, I am Surajit Malakar, a research scholar of Center for Genetic Disorder under the supervision of Professor Parimal Das, Banaras Hindu University, Varanasi, India. Today, I share my thanks, gratitude to the Students' Journal Club and the Networking Program, Session 24, for selecting me as a speaker for demonstrating, for discussing about my topics on this platform. And second of all, I thanks Buddhadev Goswami, one of my childhood friend. With his inspiration, with his help, I am here. And I also thanks the entire team of TIGS who provided me these facilities, these internet facilities, this beautiful whatever I call. Uh, Thank you. So let us enter into the topic. Am I audible? Am I
Ye-
audible?
Yes, you are.
Yes.
Yes, you are.
Or yes, yes. So
Yes,
my
sir.
topic, which you find that exploring genetic basis of neurodegenerative disorder, taking Parkinson disease as a model. So from that topic name itself, we can get some idea of the talks which we have to do for the last 1 hour. That is the genetic basis, neurodegenerative disease, and the Parkinson disease. Model means as an example. So exploring the agenda of my topic is why neurodegenerative disease, why Parkinson disease, why I choose this neurodegenerative disease and Parkinson disease as my focus of interest for giving for my research study and my future aspect and plan. And second, its importance on genetics, importance of genetics on PD. Why Parkinson disease? Why genetics is so much important for our Parkinson disease? Not only Parkinson disease, for each and every disease, genetics is so why so much important. And second, our research overviews. My research overview. So from this graph, we can clearly visible that there are three types of disease. Most effective, most deleterious disease. One is cancer, neurological disorder, and respiratory disorder. In this graph, we see that in this 2010, neurological disorder, the graph of neurological disorder is increasing. And in 2010, there was one paper that on the next occupational epidemic, occupational environmental medicine paper from the journal Occupational Environmental Medicine. It's told that globally neurological disorder accounts for about 4% of all deaths and about 5% of the disability adjusted life year for non-communicable disease. But there is no diagnosis and very little cure. In latter, in 2020, this neurological disorder leading cause of disability and the second leading cause of the death worldwide. Now it come to the second leading cause of the death worldwide. And in the past 30 days, for past 30 years, the absolute number of death had increased to 39%, and the disability adjusted life years to the have increased by 15%. It is published in Lancet 2020. And another article was published in 2023 in Clinical Medicine, Journal of Clinical Medicine, that neurological disorder are the leading cause of physical, cognitive, and disability across the globes, currently affecting approximately 15% of the worldwide population. Approximate absolute patient number have considerably climbed up to 30 years. So from all this data, these are the some few data. We have all other papers reference as a reference. From this, I gain an interest that neurological disorder is gaining its importance to be studied and to be focused about for making any medicine or some remedies for a person who are seriously affecting by this disorder. And secondly, up to this mark, there is no probable cure for this disease. The only medicine which we get that only deprived, or what we can say, only that subdue the symptoms of this disease up to a certain period of time, certain age, certain period of age. After that, the disease prevailed. So there are a neurological disorder can be divided into different categories. Some of neurological disorder is basically means a disorder which is associated with neurons, our brain, our spinal cord, central nervous system, peripheral nervous system, any types of disorder which associated with our neurons. It can be a tumor. This actually this classification is based on the symptoms and the affecting areas. Like one is tumor, seizure disorder like epilepsies, disorder of development. This includes like development neurological disorder like ASD, autism, and another is degenerative disorders, and another is headache and pain. It is very common, headache and pain disorder. This neurological disorder, which is called neurological degenerative disorder, is very important because it constitutes the major portion of the neurological disorder. About 80%, about not 80%, about 40% of the neurological disorder case entered into that neurodegenerative disorder. So that neurodegenerative disorder, according to our literature survey, annual incidence rate of 10 to 15 per zero point 0.1 million, 0.1 million person in most of the surveys, and the prevalence may rate to 2% in the person at the age of 65 years and older. This prevalence estimate in an Indian population, uh, a survey was done in 2009. This prevalence was estimated at 3 to 11 per thousand of the population, and the incident rate from zero point 0.2 to 6 per thousand of the population per year. It was published in Lancet 2021. So this major contribution from the rural population, it's the most important part of this. One is urban population, another is the rural population. We see that a rural population is mostly affected by various neurological disorder than the urban population. So there might be some cause due to the rural population. This rural, and if we see that where is this rural population is, we found that it is mainly in the Indo-Gangetic plain, that is in the east of UP, Bihar, West Bengal. This population was found to be mostly affected by neurodegenerative disorders than most of the other Indian populations. So what is neurodegenerative disorder? In briefly, we can say that neurodegenerative disorders is an umbrella term. It's a neurological disorder. It's an umbrella term which associate with the progressive loss of structure and function of the neurons, including the death of the neurons. And we all know that once a neuron die, it will not generate in an entire period of life. Excessive death of one and more population of the neuron can cause different neurodegenerative disorder. There must be various etiologies that must associated with the neurological disorder, like misfolded protein and abnormal protein accumulation within the neurons, lysosomal depletion, mitochondrial dysfunction, impair RNA homeostasis, alter glial function, and inflammation. These are various types. These are various common, not a types. They're better to say various common types of etiology which is associated with neurodegeneration and cause a death of our neurons. So we tell that there are different types of neurodegenerative disease. One is Alzheimer. Most of the four most common neurodegenerative disease are Alzheimer's, multiple sclerosis, that is ALS, amyotrophic multiple sclerosis, Parkinson disease, and Huntington's disease. This study was done on 2019 on US prevalence of selected neurological disorder. And very often, like previous case, we found that Parkinson disease is the second most common neurodegenerative disorder among all other neurodegenerative disorders. after Alzheimer disease. And according to the US study, its prevalence is 1.5 million pupils. So, what is Parkinson disease? So, Parkinson disease is primarily a progressive degenerative disorder. It was first identified by James Parkinson, and it mainly caused due to the progressive death of the dopaminergic neuron in the midbrain of the substantial nigra compactor region of the midbrain. And 1 to 2% of the population over the age of 65 years are found to be affected, and the prevalence of this disease increased by 4% in those who cross the age of 85 years. And 8% of the Indian population is being affected by Parkinson disease. Now, this Parkinson disease if we see that Parkinson disease, we found that a very interesting ratio between male and female, where male are more affected than the female. Male in a ratio of 3 is to 1. 3 is to male is to female ratio is 3 is to 1, which is affected by Parkinson disease. The pathological hallmark of PD are loss of nitro stratial dopaminergic neurons and presence of intraneuronal proteinaceous cytoplasmic inclusion, which termed as Lewy bodies. This is an pathological hallmarks. During the brain dissection, we can find that of a brain dissection of obviously a death patient, we can find a Lewy body present within the brain. So if we see that what are other symptoms, which is clinical symptoms, which is related to Parkinson disease, one is rigidity, another is tremor. This tremor is unilateral tremor, and bradykinesis and postural instability. With the etiology like oxidative stress, genetic predisposition, abnormal protein processing, mitochondrial abnormalities, and environmental exposures. Then different depending on their various types, Parkinsonism disease, later better to say Parkinson disease, can be differentiated in various types, can be classified in various types. One of primary classification of Parkinson disease depending on its cause is primary Parkinson, atypical Parkinson, multiple systemic atrophy, dementia associated with Parkinson, of which this primary Parkinson is a classical Parkinson, which is called the idiopathic Parkinson. Now, idiopathic, in a brief terminology, we can understand a Parkinson without any cause. It might be some genetic cause, some environmental cause, might be some external ingredients like some pesticides, insecticide, which may regularly ingested, and that may cause a damage to our dopaminergic neurons and may lead to the Parkinson disease. Even more than that, depending on the age of onset, Parkinson disease is defined, can be classified into three types. One is juvenile Parkinson. These juvenile Parkinson cases are most of the case which is found before 20 years of age. These patients are mainly before 20 years of age. Another is young onset of Parkinson disease. This young onset of Parkinson disease are the disease that patients patient are affected by Parkinson between the age range of after 20 years to 50 years. And after 50 years, the Parkinson disease which was affected, we can say that which is affected, we can say it is an late onset of Parkinson disease. So the most severe, it is the juvenile onset of Parkinson disease. First of a case, it affect that juvenile stage in a patient, and second, its symptoms are more devastating than any other Parkinson in other types. So when we state about idiopathic Parkinson disease, we can focus different types of environmental effect, genetic effect, and some external ingredients effect. So from genetic effect, there is a relationship with the Parkinson. Now the genes have some relationship with the Parkinson disease. Over a past decade, the genetic basis of rare PD form with Mendelian inheritance was found, represented no more than 10% of the cases has been investigated. More than 16 loci and 11 associated gene has been identified. Of these 16 loci and 11 associated gene, still now there are 5 important Parkinson disease causing genes are there. They are SNCA, LARC2, PARK2, PINK1, and DJ1. You can all see the inheritance pattern of this disease. SNCA and LARC2 inherited in autosomal dominant mode, whereas Parkin, PINK1, and DJ1 is inherited in autosomal recessive mode. Even we can found the chromosomal position, gene size, and number of exon. And interesting to state in this part is that SNCA is very less amount found in an Indian population. In Indian population, we uh, couldn't find SNCA in not so much amount. It is a very less in number, SNCA. Whereas in LARC, it is mainly found in the old onset present, and DJ1 also found it old onset present also, but PINK1 and Parkin, it is a most prevalent genetic cause for Parkinson disease in Indian population, and it is found in the juvenile case as well as the young onset present. So my interest is on this PARK2 and PINK1. So let us enter into some of the our work which was done in a lab. This work was done by my former research scholar from our lab, Dr. Tamali Haldar. It is depend on novel P10 induced putative kinase PINK1 variant in Indian population, Indian Parkinson disease patient. So actually in this case, 114 patient was screened on PINK1 gene for was screened on PINK1 gene, of which 1% was found to be carrying a novel variant that is at a 799 in cDNA position of PINK1 C2T. It's a nonsense mutation and cause a stop codon instead of glutamine codon. There is the exon 4. We can find it. This is the structure of the PINK1 gene. It is a membrane transmitting site, and it is the kinase domain where we found that exon 4 here is the affected of the novel mutation, and this patient of course is young onset Parkinson disease patient which is 35 years old, and he has the disease of 16 years. Disease duration is 16 years, means he has disease in 20 years of age with tremor in body, bradykinesia, postural instability, rigidity, and but there is no dopamine response, L-DOPA response. That is the only medicine we used to give for that Parkinson disease cure. So the experimental setup, like we had collected the human blood samples from Sir Sundarlal Hospital, IMS, BHU. There was total 114 sporadic patient sample. This 20 to 5 ml of peripheral blood was taken, and DNA was isolated from the whole genomic DNA was isolated from that blood and enter into the screening through Sanger sequencing of the known and unknown variant, and then we undergo data analysis, that is in vitro data analysis. In vitro data analysis, we had done site-directed mutagenesis in PINK1 cDNA construct. We all know that a mutation or the variation of our interest is being inserted into a clone of our gene, that is through an artificial process which we called a site-directed mutagenesis system. And later cell culture was done. COS7 fibroblast cell line was maintained in DMEM with 10% FBS solution, antibiotics, and growth in 5% CO2 and humid incubator. And second, transfection of the wild type, both wild type and mutant plasmid was done in a 1 million of cell population, and protein isolation and Western blot was done to demonstrate whether there is a functional about the functional change occurred in that protein or in the due to that mutation or not. So here in the result part, we can see that this is the wild type wild type wild type sequence, and here it about the mutant sequence, that is a patient sequence, and we find there is a C10 chain in the exon 4 of the PINK1 gene, which is transferred to a T amino T base, that is a thymine. And later we had done that multiple sequence alignment on the evolutionary conserved kinase domain of human PINK1 and with its orthologous position at novel nonsense variant. We found that it is mostly conserved. This Q, means glutamine, is mostly conserved in all of the species, in various species like pan, Homo sapiens, cow, etc. So after Western blot analysis, we found this is the Western blot analysis result. This 42 kilodalton band, which we are seeing that it is a beta actin, that is a housekeeping gene, and another uh, is that mutant. Another is the wild type, that is 100 kilodalton protein. After Western blot, we found that this, as the from a nonsense variant within that gene, it from a truncated protein of 55 kilodalton, which is easily seen in this Western blot analysis. So we can say up this potential pathogenic variant was identified in kinase domain of P10 induced putative kinase of pink1, and this variant is found in a sporadic case and cause a truncated protein, which may cause the disease phenotype. But this is the only a part of our study. We are doing others type of research, which we will have discussed in the next slides. Sorry, next discussion. Another experiment was done on a sporadic cases. Similar type of methods was followed, but data analysis was done in silico part, not in vitro. Till in silico analysis was done. So that screening was done on a pink1 variant of a young onset patient, which his age was 35 years old, male patient. And they are interestingly, they are found an mutation in a heterozygous condition at exon position 10, and two SNPs, one at similar exon 10, another is exon 6. So this is the condition of the gene within a same pink1 gene, but sorry, Parkin gene. We found a three variation, one mutation in heterozygous condition and two SNP in homozygous condition. Then a question arises: what is the effect of the SNP on the mutation? Does this SNP, which is known to be uh, non-deleterious, has an effect on this mutation or not? So in in silico prediction of variant, we had done with a various softwares, like first the depending on the software like PD, SNPs, Meta SNP, ASN, AP, Panther, Shift, and Polyfen, to find whether it is deleterious, what is the deleterious score, whether the whether the variation is disease causing or not. This we had done with a three variant, where we found that the mutation which in heterozygous condition found in exon number 10 in the Parkin gene is found to be disease causing in every software except PDSNP, whereas the other two variant, which was found neutral in most of the case except SIFT and SNP. After that, we had done 3D structural analysis. 3D structural analysis, we found that 3D structural analysis was done but in grouping in a group like structure in a specific position, one with wild type, another structure with mutation, that is 366Q. Another structure analysis was done with mutation plus both of the SNP. So here we found that this structure, which contain mutation plus two SNPs, this structure is more different, is more different than that of the wild type, and the structure containing a single mutation. From this, we gain interest for further studies like mRNA structure study. It is a very interesting result. We found that in mRNA structure studies, this is the wild type mRNA fold, and this is the mRNA fold having the mutation, whereas this mRNA structure which we found is contain mutation plus two SNPs. Here we see that there is a structural change in this position of the mRNA fold, which was not found in mRNA, this mutant, as well as the wild type. So, and this delta G value, which is free energy value, is increased in case of that this mutation plus two SNPs variant, which shows that there must be some problem relating to SNPs with the mutation. Then we draw the polar plot. Then we plot this variant with polarity index, depending on the that type of with three types of protein. One is wild type, one protein with that mutant variant, another the mutant variant protein with two SNPs, where we find that a change in this polarity region, polarity plot, in cases of this, when these three variant are together, rather than this wild type and this wild type and this mutant variant. Similar result was also seen by hydrophobicity plot, where hydrophobicity index also shows a significant change in some of the region. It is more found in this variant containing that mutation and to SNP rather than this wild type and this mutant variant. Now went for an active site prediction. This active site prediction is done on whether to find whether there is change in active site region or not. So these three proteins were taken. One is wild type, another one mutant type, another mutant type with two variant. Here we found that there is a very much change in the active site position as well as the volume of the pocket, means active site pocket. Whereas this pocket size increases more in cases of this type of variant rather than this wild and this mutant variant. So from this, we can conclude that the two SNPs, which are known as SNP, have no role in disease pathology up to date. But when it merge with the mutation, it cause with the missense pink one variant observed in a study. This cause a deleterious. It might cause the predicted to be cause some deleterious role in the associated with our disease phenotype in producing a misfolding protein of lower stability for the response of cellular stress, leading to Parkinson's disease pathophysiology. So from the whole of this tragedy, we can state that our future aspect uh, to find any whether any genetic roles to find the genetic role related to this IPD, that means idiopathic Parkinson disease. This because very less we found, more than epigenetic studies and other studies are still left, which we have to do. And second, for generation of patient specific model system for further functional study and personal drug development. So in the last slide, we acknowledged all the patient and healthy volunteers and faculty and staff of Center for Genetic Disorder, uh, Dr. Deepika Joshi, our active collaborator, Dr. Vivek Sharma, another our active collaborator, Department of Zoology, and past and present research scholar of Parkinson, this Porimal Das sir's group. This our group and our funding agencies, and I thank you.
Thanks.
My slide is visible because one question is that polarity index plot. Actually, our proteins have different polarity. We know that there are various polarity according. Some proteins are non-polar and some proteins are polar. So after that mutation, we try to find whether this change in the molecule, polarity of the molecule might change or not. So that uh, if the molecules is polar, it is readily soluble in the cytosol, can enter into any uh, organelles, like it enter into the nucleus and can cause these functions. But when there is a change in polarity index, there must be a translocation part, uh, which it affect the translocation of these molecules to any part of the organelle within the cells. This cause oxidative increase the oxidative stress within the cell and cause the death of the cells. That's all with the polarity index. How do you analyze the variant after separation of DNA? Okay, after the DNA isolation, we done PCR of that particular region, particular exon or particular gene, and after PCR, we screened it by through Sanger sequencing. It can be easily obtained from a software that is RNA, mRNA fold. You can get it. Active site prediction software is there. You can, if you type it online free active site prediction software, you can get that active site. Every software is free. You know. Uh, yes, we are trying to search that any epigenetics modification, but that is under our research.
Surajit, I have a question. Like,
Yes.
so it's first of all a nice talk. Second,
Thank you.
and so Parkinson disease is not something like a single gene disorder, right? Like sickle cell.
Yes, yes, yes,
It,
yes, yes, yes.
so which exons do you actually target, or which chromosome numbers do you actually target and find? Like, is it
Actually, uh,
or you
mm,
do whole genome sequencing to find like
mm,
what? So there could be multiple changes in the sites, right? It's not a single
mm,
gene disorder.
mm,
So like, every day somebody is reporting some mutations in the health site.
mm.
So how you came to that top 5 one, and why not others? Like there could be many other chromosome that could be involved. Yeah.
Okay. Okay. First of all, according Parkinson disease nowadays, previously Parkinson disease was called to be monogenic disorder, but nowadays Parkinson disease is a polygenic disorder. There are number of gene associated with Parkinson disease. First, for that, we first, after isolation of DNA, we undergo a whole exome sequencing or whole genome sequencing. After whole genome or whole exome sequencing, we enter into if we find any variation in any of the gene, any of the region, then we validate it through Sanger sequencing. After Sanger sequencing, uh, we will do any functional analysis so that we can prove that yes, this variant is some deleterious to our population or not. And another case, there are some of the known variant, known gene, which is well established that they cause Parkinson disease in a patient. So for that reason, before going to that whole exome sequencing, we enter into a stage where we do that Sanger sequencing, validates Sanger sequencing screening after of that known gene, and if we get anything, then we uh, do the next functional step.
So that's come to my next small question. What proteins does it change? Like you are changing the gene, but what proteins does it change? Is it change any proteins
Huh,
that
uh,
so this mRNA got changed? It caused any change in the proteins that cause the neurodegeneration, like prions does to the
we are
neurons? Yeah.
we are still in the process of research whether this function of these SNPs and with this mutation cause any dramatic change in a protein which cause some downstream effect like that. Up to that prediction modeling, we found that there is some this cause a change in active site of the our molecule, that our pink one molecule active site change portion of that active site. Um, so we predicted that it might cause any deleterious effect in a human.
Uh, there is a question, and there is a question in the chat box. Yeah.
how I am interested in learning more about how neurogenetics can contribute in curing regenerative disorder. Yes, you can do some modeling uh, of structure, modeling of some proteins. There are various papers you can get reference from there, which type of work you want to do. Of course, you can do as an undergraduate student on in silico, because in silico research, mostly if you know something, you don't require any type of lab facilities or something. You have laptop. If your laptop system, internet system is good, you can do it. Hydrophobicity index. This hydrophobicity index means a hydrophobicity of the protein. How the protein molecule is present in an cell environment. This always, actually, this is the very least part of my topic. Hydrophobicity index, that means a protein, how the protein function within a cell environment depending on its hydrophobicity property. So depending on that, we made some graph where we found that there is a change in a graph. So we can say that the hydrophobicity property change, that means that protein have some function which is different from its wild type function. In silico gene analysis, you first read a paper and get some variant. You can already get some variant in the paper. There you can start with structuring. You then after structuring, you can do what called the uh, simulations. These things you can start from there. After that, in mature portion of your study, you can go for a drug analysis and drug development part. Genetics is the strength of my study. Mhm. Yes, yes, there are many studies out there. You can find many studies are there associated with amyloid plaques and accumulation and dopaminergic neurons. Many studies are there. And it is, I am very happy to say that my sir is there, Professor Parimal Das. If he give any suggestion to us, I will be very thankful to you. Sir, please give any suggestion to all of us. It's already done. Various studies are done whether Parkinson disease, in which part of the brain Parkinson disease is affecting. So from the MRI scanning, you can find that it affecting the midbrain region of our midbrain region, and which part is affecting. You can find out white spots and that. Through MRI studies and CT scan, it can be easily done. And other of course, some other neurophysiological proper instruments are there in IMS, means medical science, which can be easily studied for.
Hi, Surajit, can you hear me?
Yes, sir.
Yeah. So I, hear all your talk, and also I found that he had some technical difficulty, but thanks to all the organizer and the student who are really running it fruitfully, I think. So that's a great effort. So my sincere thanks to them. And it's really a good habit to culture these kind of things and should be continued because that gives a very good platform to speak out, find it, and then to see that where you are, and the suggestion comes from all the speakers or the people that are listening to you, especially even the young staff of different specialties. They're coming and asking you the question. Obviously, keep on practicing these things, increases your depth of knowledge, and also help you to find out the scope of interdisciplinary, even the transdisciplinary research, which is possible nowadays, which is not possible earlier. So that's my say and then suggestion to all the students, faculty, or the postdoc students. They're listening to that. That's all at this moment. My suggestion is, yeah, good.
Thank you, sir.
Mhm.
Thank you very much, sir, for these enlightening words, and thank you very much, Surajit, sir, and also Buddhadev, sir, is here. He was a speaker in our previous session. So
Yes.
also thanks to all the participants and uh, for joining this session.
Thanks everyone. Yeah.
Thank you very much to all. It's my pleasure to join there and can speak with all of you.
And we are coming up with these sessions every week, so I encourage you also to share with these with your students also, so that it can, maybe increase the reach of this initiative, this great initiative that all of us are taking. So I want to like expand the collaboration with you guys also, so uh, we can connect afterwards. And also, if any students they want to ask their doubts, like follow-up doubts with our speaker, so they can just write to me in the group, or they can just contact anyone at Biopractify. So we will connect with you with the speaker. So that's all from my end. Thank you, everyone.
Thank you. Thank you, all my listeners. Sir, this organizer, my sir, for giving such a time to me. It's my pleasure to introduce my talk to discuss with you about the matter. And secondly, I sorry for the inconvenience for the late, which I should not do it, but I had done due to some problem. Thank you all.
Thank you.
Bye bye.
Uh, so we can disconnect.
Yes, yes.
Okay.
Okay. Thanks you. Thanks. Bye.
One thing, you can get
Yeah.
my ID from organizers,
Yeah, I will
email
share the
ID.
email ID with the
Huh.
participants. You come up with your
Uh,
doubts, and I'll share your doubts with with sir.
okay. Okay. Okay.